Hello, I'm Luna Natoli, a graduate student from Cornell University, and in this video, I will be talking about heat stress in plants. In plants, heat stress occurs when temperatures rise above a plant's heat tolerance threshold, the temperature a plant can withstand without experiencing significant and irreversible changes to growth and development. Plants have adapted to a variety of climate conditions around the world, resulting in the development of different heat tolerance thresholds. For example, broccoli, a cool season vegetable, has a lower heat tolerance threshold than maize, a crop adapted to warm, dry climates. Rising global temperatures have made heat stress a subject of increasing concern in recent years. The rate of global temperature increase has accelerated significantly in the last 50 years. Global temperatures are predicted to increase exponentially in the future, although policy reform regarding fossil fuel usage could significantly slow the rate of global temperature rise. Increasing temperatures are expected to shift the suitability of arable land for major agricultural crops, resulting in decreased land availability for some plants and increased suitability for others. Only a small percentage of the world's edible plants are being utilized in agriculture at present, and many of them are sensitive to temperature increases. Agricultural practices are focused on cultivating about 0.3% of the edible plants on Earth. Recent research into heat stress has put an emphasis on reintroducing heat-tolerant genes from wild species into our domesticated plant varieties. Cultivated carrots are susceptible to heat stress, but recent research has identified wild carrot varieties capable of withstanding high temperatures. But what does heat do to plants? Heat stress leads to discoloration in leaves and eventually leaf drop. It reduces stomatal conductance, affecting the plant's ability to respire, deactivates enzymes essential to the plant's metabolism, decreases water uptake, and results in the formation of reactive oxygen species, harmful molecules that disrupt the plant metabolism and cause oxidative stress. Plants will attempt to mitigate heat stress by activating stress response pathways, redirecting resources and energy away from other important functions like photosynthesis and respiration. Because of the insulating properties of soil, plants generally experience high temperatures in their shoots and leaves rather than their roots. When energy from the sun is absorbed by plant leaves, some of that energy is converted into sugars through the process of photosynthesis. The remaining energy is emitted as long-wave radiation or dissipated by evaporative and convective cooling. Plants have developed several unique physiological strategies to regulate leaf temperature. In the desert, some plants have elongated epidermal cells, resembling white hairs on their leaves and stems. These hairs, known as trichomes, reflect sunlight to prevent overheating when temperatures are high and provide insulation when temperatures drop at night. Some plants reorient their leaves to reduce the surface area exposed to sunlight during the hottest parts of the day. This adaptation is known as periheliotropism. Other plants will attempt to escape heat stress by speeding up reproduction, investing their resources in the survival of their offspring by prematurely flowering or bolting. Many leafy vegetables will flower prematurely when faced with temperatures above their tolerance threshold, resulting in reduced yields for crops like kale, lettuce, and chicory. While soils maintain cool temperatures below ground, solar radiation can heat the soil surface, exposing plants to radiant heat from the substrate below their leaves. These conditions can induce thermomorphogenesis in plants, a condition in which plants will elongate their petioles and orient their leaves away from the soil to avoid the heat. Temperature increases can disrupt plant photosynthesis, the production of sugars from CO2 and sunlight, but some plants have adapted to photosynthesize under high temperature conditions. High temperatures contribute to increased evaporation, impacting plant transpiration rate. When temperatures rise, plants close their stomata, epidermal pores that regulate gas exchange, to conserve water. Stomatal closure reduces gas exchange and CO2 accumulation, leading to a suboptimal CO2 and oxygen ratio in plants. Stomatal closure traps oxygen in chloroplasts, inducing photorespiration. Rubisco, the enzyme responsible for incorporating CO2 in the Calvin cycle during photosynthesis, can preferentially interact with oxygen instead, resulting in a net loss of fixed carbon through the wasteful process of photorespiration. Some plants have adapted to resist and even alleviate photorespiration, reaching their peak photosynthetic rate at higher temperatures than less heat-resistant plants. Photosynthesis in C3 plants is inhibited at high temperatures, which instead induces photorespiration. C4 and CAM plants have developed anatomical adaptions that prevent photorespiration under high temperature conditions. In C3 plants like rice, wheat, potatoes, and most trees, CO2 enters through the stomata and is fixed into glucose by Rubisco. For C4 plants, specifically adapted bundle sheath cells help to deliver CO2 directly to Rubisco. The enzyme, a phosphoenol pyruvate, fixes CO2 into oxaloacetate, a 4-carbon molecule which is then delivered directly to Rubisco, preventing photorespiration by limiting the exposure of Rubisco to oxygen. Desert plants like cacti photosynthesize using the crassulacean acid metabolism or CAM pathway. They have adapted to open their stomata at night to minimize evaporation. CO2 absorbed by the stomata is converted to organic acid by PEP carboxylase and is then stored and converted to glucose during the day. This pathway similarly mitigates the interaction of oxygen with Rubisco, preventing photorespiration. Beyond photosynthetic adaptions, plants have developed molecular heat stress responses that improve their overall heat tolerance. When plants experience heat stress, cis-acting heat shock elements will recruit trans-acting heat shock transcription factors to generate heat shock protein mRNA, single-stranded RNA instructions for the synthesis of heat shock proteins. Heat shock proteins facilitate the refolding of protein that has become unfolded when temperatures rise. They also prevent protein aggregation and help to maintain cellular integrity. Heat shock proteins are constitutively expressed, meaning they are always present in the plant, but heat stress rapidly triggers upregulation. Other heat stress responses in plants include the production of antioxidants to counter harmful reactive oxygen species and osmolytes to stabilize proteins and maintain cell turgor. Plants have adapted many strategies to counteract heat stress and to thrive in different climates. Understanding heat stress and plant thermotolerance is key to adapting agriculture to rising global temperatures. Thank you for watching.